We go. It didn't do the sound, so I was like, is it recording? But there we go. All right. All right. So welcome to Science Cafe. My name is Julia. I'm a PhD student in Neuroscience and Behavior program at UMass Amherst, and I will be the MC today. Established in 2011, Life Science Cafe is organized by graduate student researchers at the University of Massachusetts Amherst. Each month, we invite different experts to discuss their research. Typically, we host these events in the Amherst community, but we are happy to continue our efforts virtually during this very interesting time. We would like to thank our funders, the Organismic and Evolutionary Biology Program, the Graduate Student Senate, and the Graduate School at UMass, the National Science Foundation, and our generous UMass Kids donors. For the next event, we will be discussing culture and safety in LGBTQ plus communities, an anthropological explanation with Dr. Elias Capello on Thursday, April 22nd. To get reminders about all our future cafes, please don't forget to subscribe to our mailing list. And sorry, I'm just getting an alert from one of my viewers that you might not be able to see anything, but you should be able to access that. I think it's okay. I apologize. So today we are very excited to have Dr. Yadilette Rivera-Colón with us to talk about all things enzymes. Dr. Rivera-Colón is currently at Bay Path University, where she's both a professor with her own lab and the undergraduate research coordinator, program coordinator. Thank you so much for joining us today. We really appreciate it.
Thank you for having me.
So to start us off, we're going to start off by taking a look and see a little bit about what our viewers might know. So we've got a little bit of a quiz for you to start off. So I'm going to pop up a poll, and essentially the question is, which of these commonly used dietary aids may contain enzymes? So I'm going to launch some polling. We're going to see what you guys think. Might be an easy one, might be a hard one. We're going to see. I think we went too easy on them, but that's okay. All right, just give you guys another moment. All right. So I think everybody got it right, I have to tell you. But I even hid the part that does say enzyme down at the bottom. So it's cool. But I think there's too many lactose intolerant people like myself that do in fact have say that down already. But yes, everyone is correct. That is the correct one. All right. So that is a type of enzyme, but what exactly is an enzyme? Like what are we talking about when we're trying to say, oh, here's lactate? What are we talking about here?
So basically, an enzyme is macromolecule produced by living organisms, and it its job is to speed up reactions. So it's a biological catalyst.
Okay. So what is it made of? If you're like looking under a microscope, can you see it? Or are you just like looking at like a little like tiny like thing that's moving around? Or like what is the actual structure kind of like?
So scientists are very clever, and we have been stealing secrets from nature for a long time. And basically, you can't see a protein with your naked eye. However, you can definitely use different techniques in order to see an enzyme. You can extract it from a living organism and put it in a gel, and then you will be able to see a little band and be able to say, okay, this band corresponds to my enzyme, assuming that you know how big it is. But I'm a structural biologist, so my job as a graduate student at UMass was to solve the structures of different proteins. And the way to do that, well, first of all, proteins are made out of amino acids. So for some of the people who exercise and decide to bulk up, one of the things that they are recommended to do, right, is to take protein shakes. And and do different amino acid supplements. So, especially the branch chain amino acids or whatever. And those are the building blocks for different proteins and for enzymes as well. Although not all enzymes are proteins, there are ribozymes that have an RNA component. So those are made out of nucleotides, but for the most part, they're made out of amino acids. And basically, for me to be able to see and know the shape and the structure of an enzyme, I actually use X-ray crystallography,
Okay.
where you take a bunch, as much as possible that you can, of this protein and condense it into a really, really put it in different conditions in order to get a crystal.
Hmm.
Kind of like you can think about, like a little grain of salt,
Okay.
right? And then that is really highly concentrated enzyme in there. And once you have that, then you can put it in X-ray. You have to protect it, of course, because X-rays would be really hot, and then you would be cooking your enzyme. So you have it in a cooling stream of liquid nitrogen to keep it really, really cold. And then you're able to use the X rays in order to actually get the locations of different atoms. Of course, in order to be able to do that, you have to do a lot of math and physics that thankfully is now mostly done by computers. And you have a user interface that then you can basically put the puzzle together, which
Okay.
that I think is my favorite part.
Okay. Okay. So I'm going to stop sharing for a minute so we can see ourselves a little better. But so when you say enzymes and we're talking a lot of different things, we know like lactate, that's important for people who have that. But what's the purpose overall of like why is it important to have all these enzymes? Like there's a lot of different types, but like what's it doing for us?
So if we have tons of different chemical reactions in our bodies, right? And they're happening every second.
Hmm.
So I think that I looked it up and it was in the order of billion the reactions that happen in a second in a human. And basically those reactions happen because of this catalyst, this enzymes being able to speed up the reactions.
Hmm.
Without the enzymes, those reactions would take so long that life wouldn't be able to happen the way that we have.
Okay.
We really need to get it going, and it has to be in nanoseconds, second scale. So that's why the enzymes are so important.
Okay. So without what are some processes we couldn't do without them? Like what's something that's like key? Is it everything or is there just like a lot of specific things that we really need them for?
Well, we need tons of different ones, right? When we're talking about eating, right? Like the first step, as you're putting it in your mouth, you have an enzyme called amylase that is going to be breaking down carbs, right? And that's just as it touches your mouth, right? So that's just the beginning. From there, all the way of extracting all the nutrients and getting it through your body, through your bloodstream, in order to get the different components that you need. It's There's tons and tons of them.
Okay. And are they're found in all living organisms, I'm guessing. Is
Yes.
there how does that compare to like other organisms? Is there a lot of crossover? Do we all share the same enzymes or is there a lot of different ones in between different species?
So basically, humans have about 1,300 or so different types of enzymes, but amounts of enzymes, there's tons in every cell. Compared to, for example, E. coli, they're about on the order of 600 different enzymes. One of the things that makes the number so different is that in E. coli, many of the enzymes can do many roles,
Hmm.
and our enzymes tend to be a little bit more specific
Okay.
for different compartments and different parts. And the more sophisticated the organism, usually the more complicated the biochemical reactions are, and you need more specific enzymes.
Okay. Okay. So how do the enzymes work? So there's a lot of different things, but like what are we looking at and what is happening when these enzymes are doing the work that we need them to do?
So, depending on the enzymes, different things are happening. In many cases, it's all about proximity and orientation. So, being able to have the molecule that you want to process in the right orientation and in the right proximity to this specific part of the enzyme called the active site, then that's where the chemistry happens. So if it's an enzyme that its job is to cut other enzymes, then in that active site, what the chemistry is going to happen is going to release as a product that cut enzyme.
Okay.
So there's many different types of reactions that can happen that way.
Okay. Okay. So they're all just different processes and each one's kind of a little bit specific within each of its reactions. All right.
Yeah.
So we can do things like lactate. We do eat those and we take that in. Are there other ones? Do we acquire enzymes or do we basically just have everything we need already innately within us?
So usually you make your own, right? But in cases like people who are lactose intolerant, they may have a defective enzyme. They may not make enough, or they may not make any at all. And in that case, consuming the enzyme helps. However, very little of that enzyme actually gets to the right place and does its job. And there are different ways of delivering enzymes. For example, there's a technique called enzyme replacement therapy, where actually a person gets an IV infusion of the enzyme in order to be able to replace the defective or missing enzyme. But for the most part, it's really hard for us to actually get a already made enzyme into the body. And that's why a lot of scientists are working on that problem.
Okay. All right. So we are going to take a break right now and we're going to see if anybody in the audience has any questions. So just as a reminder, you can either raise your hand if you want to ask a question verbally and we'll unmute you, or you can put it in the question and answer down at the bottom and type that in and we will answer that as well. And we're going to give people a second because it sometimes takes a little bit to
Yeah,
type in the question.
typing.
I am going to ask a question as we get started. What sort of enzymes would you replace using IV? Is there like specific types that you'd be able to do?
So actually, when I was in UMass, the enzymes that I was working with were lysosomal enzymes. So in order to make it to the lysosome, you would think, oh my God, it has to go through the cell membrane and it has to be able to make it to the lysosome. But there's actually a system that is already ready for uptake, which is through a carbohydrate that it's in the cells, and then being using that as a, as usual, stealing from nature and being able to get that process kind of hijacked.
Mhm.
You can use it and then put it in the bloodstream, and then the cells themselves will be able to uptake those enzymes and put them in the lysosome.
Okay, okay,
But it's not as easy for other things.
that's fair. So we do have one of our first questions. So do the enzymes in plants differ significantly, or in what way do they differ from like animal enzymes?
So if you actually look at them, they're not that different. um, Many of them have different domains, especially active site that has all of the elements to be able to do the chemistry. Those tend to be pretty conserved throughout different living organisms, even all the way to bacteria, yeast. But some of them, if it's something that is very specific to a specific organism, you might not find it in plants.
Okay, okay, cool.
Or vice versa. You might not find it in humans and find it in plants. We don't make our own food, so.
That's fair. I wish we did sometimes. All right, so another one is what's the proportion of enzymes? What proportion of enzymes to the substances they are catalyzing? Like, what do you need? Like, is there a large amount that you have to have, or do you just have a little bit of enzymes and a lot of like other stuff? Like, what's that look like?
So it's different for different enzymes. So there are enzymes that work together as multiple copies of itself in order to be active. Some enzymes are one to one, right? Like one enzyme to the substrate, but the enzyme actual concentration is actually pretty low
Okay.
because enzymes are fast and you can keep reusing the enzyme until you no longer need it, and then the cell will make its signaling in order to be able to get rid of it. So you don't need a lot of the enzyme in order to do its job.
Okay. And then for the enzymes, do they need to be below a specific like molecular weight to be like orally taken in or available? Or is it like, does that matter? It
It
looks like it
matters. It matters. There's this rules actually called the Lipinski's rule of five for bioavailable drugs that you can take orally. and it's in 5 orders. So 500 is the molecular weight of like the cutoff for oral drugs that you can take. So in terms of enzymes, enzymes are much more complicated, right, than just a regular small molecule that you can make. So the thing is that we have a really robust digestive system, that one of the key purposes is to break down proteins as you're eating them in order to get all the amino acids. So that's why it's so hard in order to be able to actually have orally administered enzymes, just because they get breaking down pretty fast.
That's actually impressive that any make it through that. Actually, now thinking about it, that we get anything at all from that. All right, so we're going to do one more question. Feel free to keep typing it in and we'll answer it in the next session. But is there any part of the body that doesn't have enzymes or doesn't need it?
That's a really good question. It's hard for me to think about a part of the body that doesn't have enzymes, specifically because there's so many chemical reactions happening
Mhm.
that, yeah, I don't know.
Okay,
But that's a very good question.
might be stumped. We can do a follow-up post and Google that and see if we can find that answer, because I like that question. All right, so we are going to be moving on to the second vignette. So we're going to be talking a little bit about what type of enzymes you study. So I know we've talked before, but what sort of enzymes do you currently work with, and what's kind of the goal of what you're doing with them?
So, if you can show the slide about what enzymes look like real quick.
1 second. There we are. Do you want to go to the third one?
Yes.
Spoilers, guys.
There we go. Yes. So this is basically what I look at, right? Of course, in real life, they're not colored. They're colored for our ease of identifying what things we're looking at. So this is basically what the different enzymes look like. These were enzymes that I worked on while I was in UMass.
Mhm.
They were all lysosomal enzymes.
Okay.
And now what I'm working on now is more acetyl transferases. And I'm going to talk a little bit more about that. So if you can go to the, the first slide that you showed.
Great.
Okay. So go back one.
Go back one.
Yep.
Oh, this one?
One more. One more. One more.
One more.
Okay.
There might be a little lag between my
Okay.
computer and yours.
Yeah, yeah,
Okay.
yeah.
There we
That
go.
makes sense. So this is an overview of what I did at UMass, right? They were the lysosomal enzymes that if you have one of them missing, it becomes a disease in humans.
Okay.
And so you have the disease in there. You can see that little picture next to disease where there are these huge white blobs, so to speak.
Mm-hmm.
Those are enlarged lysosomes
Okay.
because there's so much of that substrate that now is not being broken down
Okay.
and it's just accumulating there excessively.
Okay.
So we know the genes that encode for those missing enzymes. We express them in insect cells
Okay.
and then purify the protein, concentrate it, make it into crystals. Of
Okay.
course, that makes it sound so easy. It's a lot more complicated than that. But once you get a good and nice diffraction pattern from the X-ray crystallography, you build the puzzle and then you have the whole structure.
Okay.
So after I graduated from UMass, and now you can go forward. After I graduated from UMass, I went to UPenn to do my postdoc with Dr. Ronan Marmestein. I should add, I did my PhD with Dr. Scott Garman. So I went to work with Dr. Ronan Marmestein, and in that case, I was doing the same thing, but in that case, I was working with acetyl transferases.
Okay.
And the expression system, although everything was the same, the expression system was different.
Okay.
We grew the proteins in bacteria for the most part or yeast.
Okay. And I can see it does also have some disease effects as well with some
Correct.
of it. So
if you're
for
missing
acetyl
every time you're missing an enzyme, chances are it leads to disease.
that's fair. I feel like they're fairly important. We've established that.
Yes.
So why study acetyltransferases over other enzymes? Like what's the draw for you there?
So part of it is the fact that We learned a ton about phosphorylation
Hmm.
as a way of turning on and off different pathways in different cells. Acetyltransferases, or acetylation, I should say,
Hmm.
has become another way of turning things on and off that people were not really studying before. But now we know that close to 85% of the different proteins can be acetylated and will be acetylated at some point.
Okay.
So what does that mean? Why is that happening? So that's what drew me to it,
Okay.
because there was so little known and it was so important. So I wanted to learn more.
And when you say turning on and off, what do you mean exactly? Like what does that mean in a cellular basis?
So basically, in a cell, you can imagine, for example, phosphorylation. A phosphate group is really negative. You can use that property in order to either prevent a protein from doing something or turn it on or allow the protein to do something.
Hmm.
So that's what it means by turn on and off.
Okay, so it's
So
essentially turning on a process.
yes,
Okay.
by controlling or modulating the behavior of a single protein within the pathway or a couple proteins within a pathway.
Okay. And so acetylation, I think I'm pronouncing that correctly,
Yes.
is doing a similar thing that phosphorylation does, which is the traditional on-off switch that we see a lot of times.
Right, but we don't know. There's
Okay.
a lot of things that we still don't know about what it actually is doing.
Okay.
But it must be important if over 80% of the proteins get acetylated at some point.
Must be happening for some reason. All right. So this is technically a family of enzymes, the
Mhm.
acetyltransferases. It's not just one, correct?
Yes,
So
correct.
what are some examples of like an acetyltransferase? Like what is like, oh, here's one picture that we could think of.
So if you go to the next slide real quick.
Hold on one sec. This one?
There we go. Yes. So this is the process of acetylation in general. So at the beginning you have just a protein, and I'm just highlighting lysine
Okay.
in because it's the site that would get acetylated. In that case, either it can be a lysine in a protein called histone, which is responsible for the packing of the DNA in order to be able to be expressed, or I mean transcribed or not. And then you have lysine acetyltransferases, which could be basically different proteins that can be acetylated.
Okay.
And there's also, and there you have the acetyl CoA and the CoA that ends up being the product at the end, as
Okay.
well as the protein that gets acetylated, which is the acetyl group is in red. And then you can also have N-acetyltransferases, which actually attach that acetyl group at the very beginning, at the N terminus of a protein.
Okay.
So they all do this um, catalysis of the reaction of taking the acetyl group and putting it into a protein,
Okay.
but they cannot do the job of each other. So each one is very specific to what it does. And even in a test tube, you can put them together and try to force it, put a ton of the substrate to try to force it to do this job, and it won't do it. So that's what I wanted to study. Why is that? What is it that within the whole structure of this protein, what is it that really makes it specific to one thing when we know that the active sites are so similar?
Okay. So what how do you do that? Like how would you try and do that? Like take a look and try and force an enzyme to do something it's not supposed to. What does that kind of look like?
So basically, what we end up doing is comparing the different enzymes within the family and choosing different parts that we hypothesize would be the parts that really give it the specificity. And once we identify those, then we try and we change them in order to resemble more the other enzyme, in order to be able to see if we can actually change the specificity from one enzyme to the other.
Okay. Okay. And so I'm going to ask a question a little bit off. So one of the examples you have in this picture back a little bit from one is this disease. What's happening here? Like what's going wrong exactly? Like is there a specific acetyltransferase going wrong?
Yes. So that is Robert's syndrome. And the acetyltransferase that's missing there is so important. It's called ESCO1, which is, ESCO2, which is establishment of sister chromatids, um, which, if you think about genetics and you think about the alignment of the chromosomes in order for the different pieces of DNA to go the right way that they're supposed to in order for the cell to divide, this acetyltransferase actually catalyzes that acetylation that would allow for that cohesion to happen so that they get together in the right place by another protein called SMC3, which is the protein that actually acts like as a little like engagement ring for the sister chromatids. And once that acetylation happens, then the DNA will go respectively to where it's supposed to go. If that acetylation doesn't happen or is defective in some way, then the DNA doesn't necessarily go the right way. And then you can imagine that that will cause a lot of mayhem in different cells.
Okay.
One of the things that you can see is just development in general gets impaired and is more noticeable in places like the hands and the feet in this
Okay.
disease.
Interesting. So a little bit more on enzymes. What is, like, maybe a little bit of a lighter question, but what's one of the favorite things you discovered or learned about enzymes in this kind of work?
Well, I'm always at the interface of different disciplines, right? Like, I'm not a true biologist, I'm not a true chemist, I'm not a true physicist, I'm a little bit of everything. And basically, enzymes have the perfect balance for me because I can see the physics in it and is true to its form. The chemistry that it does is quantifiable, right? Like, you can actually get the quantification of everything. You know how much enzyme you put, you know how much product you're making, and they, for the most part, behave compared to, for example, a cell. Cells are very temperamental, and going anything higher than that, forget it. Biology is really messy. So I like enzymes because I understand them and I can manipulate them to do different things very easily.
Okay, that's a good answer. I like that. All right, so we're going to go a little bit into kind of how you do a little bit more of what you do. And one of those things is getting a little messier and looking at yeast. But before we go to that, we've got another poll. I'm going to stop sharing for a minute. And if you were paying attention earlier, one of these was mentioned, so you will know one of these answers. But my question to you is, what famous enzyme is associated with yeast and specifically with baking bread? So I'm going to send out this, and one of those you've already got the answer for technically. So we'll see. I'll give you a couple seconds. I'm worried that I threw them off a little bit though. Though through the pandemic, a lot of people have picked up baking. So we're going to see. I tried to be kind. All right. I'm just going to give you guys a couple more seconds. All right. So most of you got it right. It is maltase. I might have thrown you off by saying that she mentioned earlier, but amylase is what happens in the mouth and that breaks down stuff immediately.
Right.
Protease breaks down protein, if I'm not mistaken, like most protein. And then lipase, I believe, breaks down fat from the name. But maltase is very good at breaking down sugars, and that's part of the baking process specifically, which, fun fact. So yeah, I might have messed you up on that, and I'm sorry if I did. All right. So my question is, why do you use yeast? Why is that like what you choose to, or if is it, does it have to be yeast in order to study these acetyltransferases?
So basically, the reason to use yeast is because it grows fast and we can grow tons of it. So that way we can make tons of enzyme. And actually, I make, for some projects that we have in the lab, we make the yeast enzyme in yeast, but sometimes we make actually the human protein in yeast. And it's much easier. It's easier to manipulate, it's easier to grow, and it's cheaper. So my lab is an predominantly undergraduate lab. So we don't have big funds. So we need to be able to be resourceful and find ways to save money. And using yeast is a really good way to do so.
Okay. How does one, is it, are you able, how does one put like a human enzyme in a yeast cell? Is that complicated? Is that a big thing? Or is it actually fairly straightforward as far as introducing enzymes go?
So theoretically, it's fairly easy because once you know how to manipulate DNA, you can use molecular biology, use PCR, restriction enzymes, being able to cut and paste a gene in a little vector that then you can put inside the yeast. That being said, to do it in the lab is actually a little bit more challenging based on the different steps that you need to take in order to do that. But my undergrads are able to do it, and they go from having zero experience in the lab to being able to express the protein in yeast, which I'm really proud of. And yeah, some students will tell you superstitions that they do of before they run a PCR, they talk to their tube before they put it in. Those things I cannot guarantee that are real, but it's only weird if it doesn't work. If it
Sure.
works, I'm gonna just let it happen.
I talk to my sea slugs. I give them pep talks before I do work with them. So I understand that. So theoretically, it sounds like technically it's a little bit like copy pasting different genes in, but the practice is kind of a situation you have to learn through a little bit than
Yes.
actually doing it. Okay. Okay. So what does a day in like research look like? Like, are you working with a lot of petri dishes? Are you working on the computer a lot of the time to look at different models? How does one work with enzymes, and what is like your specific research kind of look like for you?
So basically, it depends on the stage that a project is in. We usually start by creating and generating our hypothesis first. So that would be a lot of computer work, a lot of looking at the different proteins, superimposing them on top of each other to see what things are similar, what's different. Once you're done with that stage, there's a lot of molecular biology, so a lot of DNA work.
Mm-hmm.
So you're going to be doing amplifying DNA through PCR or running gels, DNA gels, and just ton of molecular biology. Once you have the product that you know that's the right thing,
Mm-hmm.
then is the yeast work. And yeast are very stinky and requires incubators, and now you're actually growing a living thing, so you're making this yeast produce the protein for you.
Okay.
Once you have tons of that, then you have to like extract it from there, so you have to purify the protein from the yeast. And for that, then now is more in the more like more biochemistry. So now you're actually using columns in order to separate different things by different properties. And once you do that, then you're running protein gels, being able to make sure that you make the right thing. And then we go to the enzyme activity. So at that point, you are living by the plate reader. So being able to make tons of different reactions in a small plate and having a place to actually read and see whether the enzyme did its job or not.
Okay. And then back to the drawing board if it didn't, and do it again,
Exactly. And then figuring out what went wrong.
as science always is.
Mhm.
So we're gonna have another break. So type in any questions that you have, or you can raise your hand and we can also let you unmute. We are going to start off with one. So once you solve the structure of a protein, what do you do with that? So once you know what it's made up with, what's that mean like for your research?
Well, the first time that you do is amazing. You're the only person who has seen what this protein looks like, and if nothing else, that is like one of the highlights of my research when that happens. But once you have it, you have to make sure that you're correct. So validating it, making sure that your observations are correct via statistics and mathematics and refinement. And then you submit it into this database where they'll also run their statistics and check and make sure that your protein is the model really fits the data, etc. Once that process happens and they publish it, now it's available for everybody who looks in the database. So now, for example, when I was in graduate school at UMass, I solved the structure of a protein that, when deficient, causes a disease called Morquio B disease. And this enzyme was actually being used for enzyme replacement therapy. And having the structure allows those scientists now to be able to see and make modifications in order to make it better, to be absorbed better, to make sure that they keep certain parts of the proteins that are essential for the function, etc.
Okay.
Other things that you can do, for example, if you're designing drugs and the drug, the target is an enzyme, having the structure, being able to see what the active site looks like, allows you to make inhibitors, right? Like you can put something in there to block it from doing its function, or
Okay.
an activator if you wanted to.
All right. So then we do actually have a question from one of her, you can see her Allison down here. So she's going to ask a question really quick. What you want to say?
Hi, Dr. Rivera-Colon. So I have a question about this like therapy, this like enzyme therapy that you just mentioned and that you've been kind of talking about. So you use the example of lactate as a way for people who don't make lactase
Mhm.
to basically have the same function of that enzyme. And so you brought up, so I have a couple questions. You brought up that other disease that people get when they don't have a certain enzyme.
Mhm.
So my first question is, have we come up with um, a drug or a medicine for those people that can give them a replacement of that enzyme so that they don't keep having those kinds of effects?
So, part of the things of using enzyme replacement is because there's nothing else that we can do. Um, Being able to give the enzyme is the best way of clearing the substrate that they have accumulated. But there are other things, that other smaller molecules that you can use. Those are called pharmacological chaperones. So they're basically ways of using a small molecule to help the protein that's already made regain its function by helping fold correctly. So it has the same shape that it's supposed to have normally. But those haven't been as successful as just replacing the enzyme itself.
I see. Okay. That's cool. So that's like an alternative kind of drug.
Yep.
Um, so my other question was, how do they make, well, I guess for both these types of drugs, how do they make them? How do they make lactate? Is it a fully synthetic process, or is it done kind of like how you're working with yeast, where it's like made in another type of cell? And how is it made?
So I don't know specifically for lactase itself, but I know that for many of the enzymes that are used in general, they are usually made in different organisms that are easier to work with. Sometimes, if it's a very specific human enzyme, you end up having to use human cells to be able to make it. That will drive up the price of the enzyme,
Hmm.
because human cells are much harder to keep and much more expensive. But you can have things that are made, for example, in pigs or different other organisms that allow you to make a ton, but is not as expensive.
I see. Cool. Thank you.
Yeah, no problem.
Muted myself while she was talking. All right, so we're gonna move on to our last vignette. If anybody has any more questions, feel free to write it in the question and answer at any time, and we'll get to it at the end with our last Q&A. All right, so you mentioned before that you are majorly at undergraduate lab. So what are some projects that your current students are working on?
So I have different students in different stages. So I have a student who's working on an acetyltransferase that is from a thermophile. And this particular acetyltransferase is a little bit more promiscuous compared to all the other ones that are so specific. So we're looking at to see if there's some clues as to what would have been the first acetyltransferase. So kind of like looking at the evolution of what the acetyltransferases would have looked like back in the day when we were
Cool.
still getting at the beginning of getting our enzymes ready. Um, there's others who are looking at the different changes. So we have a project that the student named um, Hat in the Cat. And it's about a histone acetyltransferase being transformed into a lysine acetyltransferase. And so basically looking at these different things in order to learn more about the acetyltransferases. And then last but not least, there's also a student looking at hypothetical acetyltransferases. So now that a lot of the different genomes of different organisms have been fully sequenced, many of them are not annotated, but we can look at the different sequences and hypothesize if this is a protein that is made, if it actually has the enzyme activity of as an acetyltransferase. So we look at the sequence, we produce the protein, and then we test and see if it's an actual acetyltransferase.
So essentially you're making like artificial kind of like creating it from what the DNA sequence looks like sort of situation.
Yes.
That's super cool. Wow. That's really impressive. And so your students' research is obviously very important to you, and you are like the undergraduate research coordinator. So what does your lab environment look like? I'm guessing it's an important thing to have this kind of like communal labs, but like what does your job look like? What does your lab environment look like?
So my lab is really dynamic. It's there's always something happening. So we have students who are working in their benches. Some of them have their headphones on and they're focused on what they're doing. Sometimes we have music on and we're all waiting for an incubation or something like that, singing and having fun. Sometimes students come just because they're studying and it's a nice, well lit place that they can get questions answered from other students and from me if I'm around, which I'm always around. I'm never in my office. I'm usually in the lab all the time. And as the research coordinator, I oversee all of the research projects that are happening. So I get to learn a ton of forensic science. I get to learn a ton of microbiology, genetics, because I have to make sure that everybody has a novel project and that is really contributing to the field.
Awesome. So I think from what you've said, it's clear that mentorship is like a big thing for you. And obviously with undergrads, they're just getting started and they're starting to work. So what kind of advice do you have for people who want to be better mentors, or what you've learned as you've picked up either from your mentors or starting to like being a mentor?
So it's really interesting because as a student, when you're first starting, you don't know what you don't know. And that's really hard, especially if you're a first generation student, like it was my case, because you don't know what you're allowed to ask and what are things that you're supposed to know. But I had a ton of amazing mentors, and I think if I saw earlier, one of my very first mentors is here, Dr. Craig Martin. And basically, I learned a ton from them. And one of the things that I learned was to listen, right? To be able to really figure out what the students are going through, and then being able to offer resources or offer solutions for um, whichever situation they're going through, whether it's in the lab or it's adjusting to the lab culture, or just the day to day of being a student. um, and also just to be able to be there for them, and then when you don't know as a mentor, then at least have somebody that you can refer them to so that they have that opportunity of continuing to seek whatever knowledge they need.
Yeah.
For the mentees though, I my advice is to have mentors of different things: academic mentors, mentors who are your peers, mentors who are a little ahead of you, mentors who are gonna be coming from behind you. So like you remember what it was like to not know, and to have that compassion and that empathy for their struggles, and being able to keep you grounded and remember that things that now you take for granted you didn't know at one time.
Yeah. 100%. As a only second year PhD, that's speaking a lot, just still in the in-between sort of area. And so how, I know that like diversity in science is a hot topic, but like, what does that mean to you? You're, if you don't mind me asking, you're a first generation scientist, and like asking those questions are hard. I know even just being like coming into it and not having any idea of how to go into college or like any of that. What does that mean to you? What does diversity in science like mean to you?
So, it's a shame that that is not recognized as often, that diversity is a strength, especially in science, when we need as many points of view as we can in order to create novel ideas and novel hypotheses to be tested. However, when you see the levels at which people study different STEM disciplines, all the way from the undergraduate level all the way to the PhD, and then getting to become professors or members in industry and many other jobs that you can have, you see the numbers are really small for underrepresented minorities. Women are still underrepresented in most STEM disciplines, with the exception of biology. And underrepresented women are even more so if we're talking about Black women who earned PhDs is around 2.2% of all PhDs awarded. For Hispanic women, also very similar. And when we talk about Native American women, it's even lower. So there are ways that we can encourage women to be in STEM disciplines. There's a lot of ways of encouraging underrepresented minorities, both men and women, and it all starts with ourselves. If we can look at what are our biases, we can work on them, right? Like if you're aware, you can work on it. And if you work on it, you will change your own environment. And as a result, if everybody's doing it, you'll change the environment for everybody. Because one of the things that happen is that sometimes we get the students from underrepresented groups, but we lose them
Yeah.
because the environment is actually not good for them to thrive. And the thing is that many of the things that you can do to encourage the students to stay help all the students, not only the underrepresented minorities. You're not taking away anything, you're just adding resources. And those resources are good for everybody. So if we could think more about ways of doing that, we will make science even more innovative and a true path for anybody who wants to pursue a career in science.
Yeah, 100%. Thank you. That was really well said.
Thank you.
And so kind of as a like a finishing up question, and this might be a little bit of a long finishing up question, but like what has been your journey to getting to this work? Like how did you get started and how did you reach this point, and like how are you here where you are today?
So it's funny. Part of it, so at the beginning, I thought I would be a nurse. So I went to a vocation high school and studied nursing there. Then during my work there, especially in the emergency room, when I did that rotation, I saw all the job that the medical doctors were doing, and I'm like, I can be that. So I decided to go into pre med for college. And of course, coming from the vocational high school, I was definitely not prepared for college at all. I didn't do any of the college prep courses. My family had never gone to college. So I had no idea what I was getting myself into. I did terrible that first year. And I questioned myself, like, is this something that I'm supposed to be doing? Maybe I made a mistake. um, my school had a program for doing summer internships, and they were all in the US. And I signed up to for one of the interviews, very excited to see if I would get in and have the opportunity to do an internship, because I wanted to, before I committed to medical school, I wanted to know if science was something that I would like to pursue. So that first interview was terrible. The person told me that I should consider a different career, that my English wasn't good enough, and I was like, "Thank you so much." And I went and signed up for the next interview. I was like, "I'm not gonna let this person dictate what I'm gonna do." So that next interview was with Dr. Craig Martin, and um, he was able to give me the opportunity to take the he took a chance on me because my chemistry grades were not that great, but he knew that he saw the potential, and he saw that I really wanted to do it, and he found a way to get me to this internship. And when after this internship, when I saw myself in the lab, I had the opportunity to do research. And my mentor in the lab, Selassie, he was amazing. He really taught me to work and do science very ethically. And I had a really great experience. So when I came back, I changed all my plans. I canceled the MCATs, scheduled the GREs, and actually took up on a research project at my school. And once you have that excitement of being in the lab and being the first to know something that you just discovered and nobody else in the world knows yet until you tell them, that to me was way better than what I wanted to do. At that point, I wanted to be a dentist. So, um, and I was actually doing work at a dentist office, and all of a sudden that seemed so boring. And it was crazy. Everybody was like, a scientist, what do they even do? Right? I never knew a scientist. I never knew a scientist. My family, they didn't know. And once I decided to go into grad school. My family, all that they could say was like, yep, she's still studying. When are you graduating? Well, this is not like regular school. Like there's more time, there's things that I have to get done. And oftentimes it was hard with my family because I didn't always have the support, mostly because they didn't understand. And when you're so stressed out, you have no energy to explain to them what's happening either. Um, but thankfully I was able to persevere. Scott Garman was amazing, a great mentor. It was a great lab to be in. And I was lucky that I had just a good support group from so many different students and professors, and just everybody was really supportive, and I had the opportunity to grow. And then, once you have done it for long enough, you gain that confidence to tell your professor, okay, I'm ready to graduate. But I knew that I wanted to teach, and that was another challenge because some people didn't understand that you're so talented, you have published, you have done this, why would you want to go into teaching? And my answer was always like, so you think that training the next generation of scientists is a low goal, something that is not worthy? And that usually have them, shut up or
Reconsider
change.
if they're
But yeah, so because I wanted to do that, I applied for a fellowship that allowed me to do both research and teaching.
awesome.
They're called IRACDA fellowships. And this one was the one that I did at University of Pennsylvania, and I got to teach at Rutgers University and Delaware County Community College, which allowed me to know the different types of teaching and the different audiences, right? Like who your students are, and then be able to really focus on what I wanted to do, and being able to bring science to different communities and different people. So once I saw the job at BayPath, which I wasn't even looking for a job at that time, I just saw it and it was back in this area and I wanted to come back. Um, I I I thought I would try. And once I went there, I was in love, and I was like, oh no, a women's college. So I get to teach women in science. Um, most of them now, like 51% of one of the classes was non-white. So I got to teach underrepresented women in STEM.
Amazing.
And when after that I was like, oh no, like I really want this job. I'm gonna be really sad if I don't get it. um I went and I talked to my advisor, Dr. Marmestein, and the conversation went from before the interview, this would be great practice for you so that you know how to go through the different interviews, to if they offer me the job, I'm gonna take it. And he was not ready. He was like, are you sure? And I'm like, yes. And then I explained, and he's like, if that's what you really want, I will support you. So again, another mentor who was very supportive. And yeah, so then once I got the call and I was gonna be able to teach, I was very excited. And one of the questions that I asked during my interview was, will you allow me to do outreach? Will you allow me to bring my science to the community? Because that's the reason why I wanted to come back to Western Mass. And if that's not going to be something that I can do as part of my job, then I don't want the job.
It's a bit
And
of
I
a
was
deal breaker
serious.
sometimes.
Yeah. And I was serious about it. I really cared. And they were like, wait, you're going to be teaching, doing research, and you want to do outreach in the community? Yes. And at the
Golden
beginning,
opportunity.
right. So at the beginning, I was a little bit like, I wonder if they're telling me this just to get me and then they are not going to let me. But it hasn't been the case. They have supported me financially, they have supported me with the time, and it's part of my promotion. It's something that they value. So that makes Bay Path a great place for me to be at, and which many people didn't understand because it's in a predominantly white town, being in Longmeadow. But once you step into the campus is very diverse. The student body is very diverse. But we're still working on making all of the parts of the campus be diverse. But definitely the student body is.
Well,
So
we're very glad you said yes because it brought you back to our area. So yes.
yes, that's what I really wanted. So I'm glad that I got the opportunity and that I still get to do this. Right, I still get to talk to the students from UMass, and normally I would be up there and we'll catch up with so many people, but of course because of the current circumstances, this is the best we can do.
Yeah, sadly, the world's conspiring us against us a little bit, but all right, so we're gonna have one last question and answer. As we're finishing up, we might run over a few minutes, but that's okay. I am gonna post just in the, I'm just gonna post in the actual chat one more time, which is just the link to the, all panelists and attendees, there we go, to the survey. If you guys don't mind taking it afterwards, it should just pop up if you click on the link in another window and you can take that afterwards. Just a reminder, helps us plan new cafes that you might wanna see. All right, so we've got a couple of questions.
Okay.
But one of them, it might be a little bit of a combination. I'm going to ask this because I think it is a good question. So what is one of the ways that you feel like the environment often doesn't support and continue to support underrepresented people? Can you give it like a good example of that? And it's a hard question, I understand, because there's a lot of complexity in that, but I think it's an important one to talk about.
So I just recently I participated in a panel about diversity and inclusion for the Biophysical Society. And one of the things that we saw, for example, for this society that is international, that has tons of scientists from all over the world, is that the percentage of scientists from underrepresented groups who get an award, they couldn't even have a number. That's how low it was. And then we were talking about what are some possible solutions. And one of the obvious ones to me was like, well, you have to nominate them to win. So even just for other scientists to recognize who is doing a great job, who might not get a nomination, right? Because they're overlooked, because they might not be friends with the right person, or they're not the first person that is thought about when something like that happens. So that's one of the which I think is the low hanging fruit, right? Like something that is very easy to do in order to encourage. Because if a student sees who's from an underrepresented group, see somebody from an underrepresented group get an award, that is something to look up to. If you can see it, you can be it. And that's something simple. Other things are the way that we um, define success, right? In an application for some school, right, for college, we might look at the system itself, might be looking at 4.0s or just high grades, and maybe a really well written personal statement or something like that, right? Like those are usually, and a GRE score. Um, we know that we've known now for a while that the GREs don't have a true relationship whether you're going to be successful or not, and they cost money. So for somebody who has financial hardship, they may not be able to do the GRE. So now that's something against them. That is not anybody being mean and trying to exclude them, but it's something that is part of the system that will automatically exclude those people.
Mhm.
Um, sometimes even just the way that they write, maybe the essay is not written perfectly, but it's a person whose English is not their first language, like that was my case. Or it can be even their third language. For some of my friends, they speak multiple languages and English is not even the first or the second. But the fact that they're able to put this application together, life circumstances. Somebody can have a really high GPA because all they were able to do and they had to do was study and be a student. Some students are moms or are taking care of their parents or their grandparents. And that's an additional hardship that doesn't necessarily make it to the application,
Of course.
that might prevent somebody from getting an opportunity in order to get in. And that's only just getting in.
Yeah.
Being able to persist, right? Being
Do
able
the
to
work and
have all the resources that you need in order to stay and be
yeah.
successful. That's another thing. So one of the things that I think that we need to work really hard is how do we define success,
Mhm.
and how to make it so that that success is actually inclusive and it's not just the system that we have been doing. And just like enzymes, right, we have to be the catalyst for that. We have to put in the energy in order to move that barrier, in order to be able to help those underrepresented minorities to be part of the decision making.
Awesome. Well, I think that's a great place to end it. That was a wonderful summary of that. So thank you so much. That was great. Thank you everybody for joining us. Our next event will be in the same format. It will be held on April 22nd. We'll be discussing culture and safety in LGBTQ plus communities, an anthropological explanation with Dr. Elias Capello. If you want to learn more, either subscribe to our mailing list or you can check on our website, which is oebsciencecafe.org. For more information. Thank you very much. And I'm going to stop recording now.